Hi, Kelly. Thanks so much today for joining me on an episode of Concentrating on Chromatography. For listeners who may not know about your work, can you tell us a little bit about your role at Thermo Fisher and how you became involved in analytical workflows for biopharmaceutical development?
Yeah, sure. Firstly, thanks for having me. It's my pleasure to be here. So, currently I'm a senior manager in market development and collaborations at Thermo Fisher Scientific. And my focus there really is on biopharmaceutical development. But at my core, I still very much see myself as a scientist. I think we all do if we've ever been there before. I did spend over 15 years in the past working hands-on LCMS and protein mass spectrometry. And so my background spans lots of things from cancer biology, quantitative proteomics, biomarker assays, and similar. And I completed my PhD in systems oncology. And then I later worked as a postdoctoral scientist at Cancer Research UK. And during both of those experiences, I applied mass spectrometry to answer very complex biological questions around drug discovery, patient stratification, and treatment monitoring. So, really, I think that scientific foundation has continued to shape how I approach my current role. Today, I work closely with the biopharmaceutical industry, so helping those guys to translate complex analytical challenges into robust, scalable workflows. And that can involve anything from drug discovery right through to regulated manufacturing. So, for me, it's ensuring that the science, whilst remaining innovative, is also extremely reproducible, compliant, and ultimately impactful for patients. It's probably not a short answer. Sorry, I just want to give you the background.
No, that's perfect. Thanks so much. And one statement I find myself making a lot is we went with the name concentrating on chromatography because my company, Organimation, we do sample concentration typically ahead of tandem chromatography mass spec workflows. But this podcast really does focus on separation science in general. So we cover a whole lot of more mass spec focused topics as well. And GLP-1 drugs have certainly become the most talked about area right now in therapeutics. From an analytical chemistry perspective, what makes this class of molecules so interesting right now?
Yeah, so they're certainly topical, as you mentioned, but I think they are interesting as well because they sit between small molecules and large biologics. So they're kind of in that gap in terms of people's expertise and types of molecules. So obviously thinking about the molecule, they're very structurally complex. They're highly sensitive to impurities and degradation. And as we know, they're being produced now globally at an enormous scale. So that combination really makes them a proof point for how modern analytical science must evolve. So, they're demanding the need for more sensitivity, for that structural insight, and that greater workflow continuity from research and discovery, characterization, all the way through to manufacturing. So, super interesting class of therapeutics at the minute.
For listeners who may not work with peptides every day, what makes drugs like semaglutide analytically challenging compared with traditional small molecule drugs?
Yeah, I think that's where the challenge does come, where you think of peptide therapeutics like a traditional small molecule when you think of it in that same kind of consideration. Peptides like semaglutide are larger than your typical small molecule, but most importantly, they're conformationally dynamic, right? And they're often now chemically modified. They do this to improve stability, half-life, absorption, for example. And being a peptide based drug, they can contain very subtle sequence variants. They suffer from post translational modifications such as oxidations, deamidation. They can be prone to aggregation. And they also have obviously low levels of impurities. And all of the above are obviously difficult to separate chromatographically, and they really can be challenging to identify. So I think all of that considered, with these therapeutics, the analytical bar is high. And it really does demand high resolution, accurate mass, and that chromatography selectivity. And all of those must work together to overcome their challenges.
And how have GLP-1 therapeutics become a proof point, let's say, for where analytical science is heading in biopharma?
Yeah, I think on a high level, what they've shown is that the future perhaps is not measuring more. It's measuring the right things earlier and measuring with confidence. Developers of GLP-1 therapies, they need those integrated workflows, but those that generate actionable insights in discovery, but then remain robust enough for that, the final phases of drug development, such as global regulatory filings. And they just demonstrate that scalable and harmonized analytical ecosystems are now becoming necessary and essential.
And peptide therapeutics, as I understand it, can contain subtle sequence variations or modifications. Why are these differences so critical to detect?
I think obviously being peptide based, that means that every single amino acid can have a substitution or a post-translational modification. And even a minor modification can then impact that molecule or that peptide's ability to bind to the receptor, for instance. And that binding or that difference can then obviously in turn have effects on potency. It can cause challenges in terms of immunogenicity, and it can affect the molecule stability. So a very small change can have a massive impact. So in that regard, from a regulatory perspective, you have to demonstrate the precise structural identity, and then you have to control for those over the development of the drug. And it's foundational to ensuring the safety and efficacy of the final drug product.
Like I mentioned earlier, I certainly find myself on this podcast and just in general talking about chromatography and mass spectrometry together in their application in modern workflows, let's say. But specific for peptide characterization, why is this combination so powerful?
Yeah, I think obviously we've talked about the need to the complexity in regards to when we compare them to small molecules. Peptide based drugs are more complex, conformationally more dynamic. So I think when we think about LCMS approaches, we think about the chromatography being able to separate the complexity, so separate out some of that complexity, which then can be detected with mass spectrometry. So really together, advanced UHPLC and high resolution accurate mass spectrometry really provide both that selectivity that's required, the molecular specificity, and all of this is required together to fully characterize a complex peptide therapeutic.
Now, in this work, specifically in your work, how does high resolution mass spectrometry change what scientists can identify compared with what might be considered older approaches?
So it's becoming an indispensable tool in drug development, but I think high res accurate mass spectrometry really delivers that exact mass, that isotopic resolution, structural confirmation, and this is all at a much higher level than perhaps another approach or an older platform just wouldn't be able to achieve. And then when we think about that in the relation of peptide-based therapeutics, it allows developers that confident identification of those sequence variants and those post-translational modifications, or for instance, trace impurities even. And it can do this no matter the matrix or the background sample. So it really provides that quality data that can stand up to regulatory scrutiny or audits and global regulatory reviews.
And I noted that one of the themes, let's say, in your recent work is detecting impurities at extremely low levels. Why is sensitivity so important in peptide drug analysis?
Yeah, I think everybody's striving for highest level of sensitivity. If we think just in terms of peptide therapeutics, the difference between safe and unsafe can really exist at that trace level. So detecting impurities, even at parts per billion levels, or identified, like I've mentioned, those really low level or low abundant variants is crucial. And it's crucial that's done early on, so that allows developers to really mitigate risk before scale up. So when we think about sensitivity and analytical sensitivity, that is required for the development, but it directly supports patient safety at the end of the day, and obviously gives you that regulatory confidence as a developer.
And how does the ability to identify co-eluting or impurities in general impact drug safety or even regulatory confidence?
Yeah, so obviously, if you can't distinguish between two nearly identical species, then you haven't fully addressed all of the risk, let's say. So I think by employing ultra high resolution mass spectrometry combined with optimized chromatography approaches, then it allows you to differentiate and clearly differentiate nearly identical species, which that takes away any ambiguity in impurity profiling. So obviously that's essential again for safety of the therapeutic and for global filings.
And one of the messages you've emphasized for analytical strategies is that they don't need to be reinvented at every stage. There isn't a need to reinvent the wheel, let's say. Why is workflow continuity becoming so important in this process?
Yeah, practically, for one, I don't think developers can afford to reinvent methods at every stage. That's wasteful. It's also, I think analytical strategies, they must transition seamlessly from research and development all the way through to QC and to manufacturing. And we've known that, we're aware of that here at Thermo Fisher Scientific, and our platforms are intentionally designed for that continuity. So at every step, we're maintaining data integrity, reproducibility, and facilitating that scalability without disruption. That's incredibly important to us as we develop those workflows.
Now, I know this is kind of a broad question, so feel free to take it in any direction, but I know that Thermo Fisher obviously touches all of these elements, but how are instrumentation, software, even data platforms evolving to support that kind of end-to-end workflow that you're describing?
Sure. I mean, it's a good point you raise. When we talk about analytical workflows, of course, here we're speaking to LCMS as a workflow, but for us, it's wider. We're always talking about consumables, the LCMS, and obviously the informatics. They're all an essential component to a workflow. But if we think specifically at instrumentation, let's say, in general, it's all becoming faster, it's all becoming more sensitive, and there's more facility for multiplexing because everybody's looking for this higher throughput. But whilst they're looking for that higher throughput, we can't sacrifice data quality and resolution. We're maintaining that. And then on the same time, if we look at integrated informatic platforms, these are now transforming those complex data sets off the back of the incredibly powerful instruments. They're now translating this information into those actionable insights and supporting our customers in compliance, traceability, and in harmonized global operations, really, these days, where, manufacturing occurs across the world.
And to stay with that in terms of modern analytical ecosystems, let's say, how do they help maintain data integrity and compliance as drugs move through the process towards commercialization?
Yeah, I think by obviously integrating validated software, security data management and audit trials, this allows for that harmonization of workflows, across sites. So it's not just about generating data, it's about ensuring that that data withstands regulatory scrutiny worldwide, and it's a secure data.
And GLP-1s are just one example of increasingly complex therapeutics. What lessons from this space apply to what you would consider next generation biologics and where things go from here?
Yeah, so you mentioned like complexity, and I think that is the major lesson. I think regardless of the therapeutic format, the lesson really is that complexity is increasing across the board. So whether it's GLP-1 peptides with have their own complexity, or more complex molecules like antibody-based structures, fusion proteins, or even more advanced biologics. The need for those deeper, molecular insights, that early risk detection, and that, again, scalable analytical control is really universal.
As patents expire and generics enter this GLP-1 market, how does analytical science ensure both safety and equivalence?
Yeah, that's again a big area at the minute as we see the patents for GLP-1s expiring across the globe. And I think, for biosimilars and generics, the burden of proof is really that rigorous structural and functional comparability. So it's not just about, well, it is just about characterization, but then we have to take that information and use it for comparability. So, high resolution analytical platforms, whilst providing that depth of characterization, this is still required for generic or biosimilar versions, but now required to demonstrate that equivalency. And we must obviously establish equivalency to protect patient safety and efficacy, and to do that with confidence and to generate generics and biosimilars with confidence, we need to ensure equivalency.
Kelly, I'd love to hear your opinion on where you see the biggest innovation opportunities in chromatography and mass spectrometry for general biopharmaceutical analysis.
Yeah, I mean, like with everything continues to evolve, and there's lots of innovation opportunities out there. It's very exciting to work for a vendor in that regard. But when we look at the biggest opportunities, I think it's greater integration, faster separations, who doesn't want that? Enhanced multiplexing capabilities, smarter data analytics, and obviously everyone's wanting more automation. We want all of that together and to maintain rigor in what we're doing. As well, alongside of all of those kind of more incremental innovations, I think another thing that we're seeing is opportunities in real-time monitoring and process analytical technologies. So that's super exciting because that's moving those insights closer to manufacturing. So again, a new area for innovation.
In this peptide work and biopharma in general, they're obviously very complex workflows. But looking forward 5 to 10 years in the future, how might these analytical workflows change and possibly become even more complex?
I mean, 5 to 10 years is a long time, but looking at the trends that we have today and the challenges, I think we'll probably see, workflows becoming more predictive, more automated, and more interconnected. We've talked about scalability and continuity, but they'll become more connected. Obviously, as with everything, we'll see a greater use of advanced informatics and AI assisted interpretation. But I think it's going to be really super important to remain grounded in high quality analytical data, and that'll continue to be generated by ultra high resolution instrumentation.
I just had a hunch that AI would be part of the answer.
Yeah.
Finally, what excites you most about the role of analytical scientists and the role they'll play in just shaping medicines moving forward?
Yeah, obviously, what excites me the most with the role of analytical scientists is the role that we play in enabling access, enabling patient access. So, when we see deeper into complex molecules and the hidden risks, not only do we accelerate innovation without sacrificing that rigor, but ultimately that powers the therapies to reach more patients safely, which is super important. And obviously, working at Thermo Fisher Scientific, our mission is to make the world healthier, cleaner, and safer. And I think, I really believe that analytical science is central to delivering that promise.
Kelly, this has been wonderful. It's so great to learn from you and really to discuss such an important and pressing area in terms of these GLP-1 peptides. So thank you so much, Kelly, to you and Thermo Fisher for joining me on this episode of Concentrating on Chromatography.
Thank you very much for having me. It's been a pleasure. Thank you.